So let's get straight on to session one, building the research landscape. As Lucy said, this will spotlight different initiatives that the Trust is involved with to build the landscape and enable research. So we've got 4 presentations, 5 speakers. One is a pre-record. But I'm delighted to introduce our first speaker, Professor Alex Horsley from the University of Manchester, and his presentation, More Than a Single Gene, CF and the NIHR BioResource. And just a reminder to all speakers, green button. Thanks, Alex.
Thank you very much, Catherine. And it's lovely to be actually physically presenting at a face-to-face meeting. So my name is Alex Horsley. I'm a professor of respiratory medicine in Manchester. I'm a consultant to the Adult CF Centre there, and I'm chair of the Clinical Trials Accelerator platform. And it's really in all 3 capacities that I want to talk to you about the NIHR Bioresource and how CF and the CF Trust are making use of this powerful national bioresource to really hopefully drive innovation and support both patients and the research community. So the starting point for this is that we all know that CF is a single gene disorder. It says so on the CF Trust website. This is your classic recessive Mendelian inheritance where you get a gene from each patient, from each parent, I beg your pardon. We also know that different patients have different expressions of disease and that of the 2,000 or so mutations in CFTR, 300 or so cause disease, and difference in the expression of these, the severity of these, result in difference in disease severity. And if that was the end of the story, then all patients with the same CF genetics would have the same patterns of disease. But for those online, patients online, parents online, and for CF clinicians, we all know that's very much not the case, that different patients with the same CF genetics can have really quite different expressions of disease. And that's because although CF is determined by the presence of 2 abnormal CFTR genes, actually the expression of CF, the final expression, is a complex interaction between genes and environment. And we know as clinicians that you can have quite a wide variation in severity even with the same CF genetics. So these are 2 delta F homozygote patients. They're the same age. One has severe expression of CF with severe lung disease. The other has very mild disease, and we see that very frequently. And that's because there are a range of different other genes that impact upon how bad that expression of CFTR defect is. And they can be other ion channels that mitigate the impact of the chloride defect. There can be other genes that affect the inflammatory pathway that's involved in inflammation and repair, and also hormone pathways that determine other comorbidities in CF. Added on top of that are all the random and environmental exposures that patients undergo. And the end result is that no 2 CF patients, even with the same CF genetics, end up having the same expression of disease at the end. And even siblings can have very different outcomes. So if this is the final CF phenotype, the final expression of CF disease, as it were, it's a combination of different genetic and environmental factors that go into determining that. And we can measure that. We know that well. We've been doing that for many years. We can make attempts to measure healthcare and compliance. We can measure what happens in the environment. We can look at stochastic events. Those are random things that occur. You know, maybe someone's had a pneumothorax or a chest injury or something. And what we haven't really paid too much attention to, perhaps, certainly recently in the rush to have new CFTR therapies, is the impact of modifier genes. And I only have 15 minutes, so I can't do this in a lot of detail, but people who are interested, there's a very good review from a few years ago by Gary Cutting looking at the different other genes that may be involved. And this figure taken from that review shows some of the different comorbidities and impacts of CF. And here on this side, showing how much of this is due to genetic variability and how much is due to environmental exposures. So the dark purple is due to variation in CFTR genotype, and the mid-purple is to other genetic modifiers. And then the sort of light grey purple is environmental exposure. And the way that these numbers are arrived at, you can look at things like lung function. You can look at twins who have the entirely same genetic makeup. You can look at siblings who have the same CF genetics, but other genes will differ. And you can look at how lung function differs between these and what happens when they have different environmental exposures. And through complex studies like that, you can discover that that CFTR genetics plays a relatively small role in determining the final FEV1. So genetic determination is around 50%, but the CF genetics, CFTR genetics, is a much smaller proportion of that. And in fact, things like Pseudomonas and FEV1, airway obstruction, the final expression is due to a mixture of environmental and genetic impacts. And here listed are some of the genes that have been implicated. And some of these are found by looking at genome association. Some of these have been discovered by seeing what's important in other diseases and looking for heterogeneity in the CF population. And you've got things like TGF-beta1, which is involved in fibrosis and repair, and BL2, which is part of innate immune response, the kind of things you might expect. For exocrine pancreatic insufficiency, so pancreas not working, that's almost entirely driven by variation in CFTR. For things like diabetes and intestinal obstruction, again, CFTR genes play a very small role, and a lot of this is determined by other genes. And then BMI at the bottom, oops, as you can imagine, is a combination of environmental and genetic things. So that's the starting point, and it's important to bear in mind that when we talk about the bioresource, we're not interested really in the CF genetics. It's all these other genes that we're interested in. And that's important because when you communicate with patients, a lot of the literature is talking about understanding the genetic cause of your disease, but we know the genetic cause of CF, we know about the CFTR, it's the other genes we're interested in. So the NIHR Bioresource is a national initiative run by the NIHR, and it's to capture the genetic causes of a range of diseases. It's run, although when I say national, it's really an England initiative, so I'm afraid the devolved nations are not, at this point in time, part of it. But there are 120 NHS trusts and around 200,000 patients and healthy volunteers already recruited. So for CF, our main interest is to capture the variation in modifier genes and understand the impact of these on phenotype. And if we can do that, that will help us understand the pathways by which these other comorbidities arise. So the process is that blood or saliva is taken in clinic, is sent to the National Biosample Centre in Milton Keynes, where its DNA is extracted and it's banked. They also store plasma in serum from blood samples too for future analyses. And then that's genotyped using a specific array developed for the biobank, which allows high throughput screening of multiple SNPs, those are genetic variation markers, and imputation of lots of other ones as well. In addition, it's a custom array, so they've added other markers for autoimmunity and inflammation. And you can ask them to run analyses for specific SNPs if you come back to them with the funding to do that and the permissions to do that. They have also completed whole genome sequencing for thousands of patients in the rare disease cohort, although that's not the standard approach at the moment. And importantly, the consent that patients give allows linkage of that genomic information back to their clinical information. So this is a summary really of where the NIHR Bioresource is at the moment. As I said, it's got 200,000 healthy individuals and participants, drawn from the general population first of all, that's the general bioresource, but also the rare disease bioresource, which CF is part of. CF is one of around 60 conditions that's part of that rare disease bioresource. And you can see there are other specific bioresources in addition to that. So for inflammatory bowel disease, mental health, liver disease, and most recently COVID. Now the rare disease bioresource that CF is part of, this was driven very much by Lucy Allen, who you heard from earlier, who has driven this process and wrote the application and has worked very hard with BioResource to set this up. And we're very fortunate that CF has been accepted as one of the rare diseases. So this isn't an open consent. Patients don't volunteer themselves like healthy volunteers can. This is consent by invitation in specific disease areas. And one of the rationales of the rare disease bioresource, and that patients will read about if they look up about it, is to understand the genetic basis of disease. And you can imagine for many patients with some of the weird and wonderful syndromes that are part of the bioresource, the genetic causes are not known or they're only partially known, and entering the bioresource may help them understand what the genetic causes may be. For CF, obviously, that's not the purpose. And again, when communicating this to patients, a lot of the literature is a bit of a one-size-fits-all, and it's important to communicate that. But it's a very powerful open research resource. So researchers are able to access this genetic information, are able to request additional genetic analyses, and are able to request recalling patients for further investigation or sampling. And CTAP, the reason that Lucy was driving this, CTAP sees this very much as an important part of the UK research offering. our ambition is to get as many patients as possible signed up and to have a powerful resource to both understand the cause of these comorbidities, but perhaps in future also, as we move away from a focus purely on CFTR, a focus on other channels, other mechanisms of disease, then maybe actually by genotyping these patients, we may be identifying patients who will respond to different therapies in the future. So just to summarize, patients who go into the BioResource provide a blood sample for DNA. They also have plasma and serum stored. They agree to a linkage with their health records, and there's a genomic analysis completed of their samples. And then researchers can access that by contacting the BioResource. You can search for patients either by specific genetics or by phenotype, and you can request access to data, you can request access to the samples that are stored, or you can request that patients be offered the opportunity to come back for further information to be gathered or for further samples. And how do you do that? Well, it's a well-worn path, so the BioResource has a number of open studies. There's a research study or a data access form that you can complete, and that's then reviewed by the BioResource Steering Committee. And they will look, obviously, for evidence of scientific justification, for statistical validation, and for evidence of benefit to patients. The approved applications are obviously subject to various checks, like ethics and funding, and then costs are charged on a recovery basis. So a commercial company will be charged costs. For academics, if it's a data access request, it's free. So how do we go about doing this? How do we get patients into BioResource? Well, the first thing is there is a 7-page patient information leaflet. And we've opened in Manchester, and what we've seen is that the information as presented is, as I said before, quite a one-size-fits-all, and important to communicate to patients that this isn't all about their CF genetics. This is trying to understand the other genetic variation, the other causes of comorbidities. There's also the CF Trust have got information on the website, and there's a video on that, which I'll talk about a bit later as well, and the NIHR BioResource website too. The consent form is a bit more straightforward. There's 4 bits to read and sign, which are fairly self-explanatory. There's 1 bit where you consent to join the BioResource and to potentially be contacted for future studies. You consent to gift your samples, to link clinical data, and to share results with the GP if that's significant. And then what we complete is a CF genetics, a CF specific data form, which is mostly just tick boxes for the CF genetics and a registry ID, so that we can potentially in the future link this back to the registry. The patient completes a data collection sheet with their contact details, and importantly, how they would like to be contacted if there are future studies for them. And then a copy of the consent goes in the patient notes. From a sample collection point of view, it's relatively straightforward. A single set of blood samples. These can be taken when bloods have been taken for other purposes, and then they're barcoded with a sample collection form, and they're put in a box and they're sent to the NIHR Biosample Centre in Milton Keynes. So there's no local lab processing, which makes things a lot easier. From an admin point of view, the consent and the data forms also get barcoded, and they get put in an envelope and sent to the BioResource Centre in Cambridge with the consent form filed in the patient notes. So there are some challenges to this, and I think we need to be open and honest about these. The first is there is no specific additional resource to support delivery. So this is supported by CTAP, and CTAP coordinators will help deliver this, but often it's a case of finding the resource from within your own department. It is an additional ask of our patients, and I'm very conscious at this time that we're not seeing our patients all the time. When they do come back to clinic, they may be getting presented with consent forms for trials, for local research, questionnaires. You know, we have to be careful not to present someone with 5 or 6 different information leaflets at once. So exactly how you approach patients will probably depend upon the centre and what else is going on. The other thing that both patients and clinicians have raised as a potential objection is this issue that CF patients may get asked to take part in other studies. And this is an explicit and important part of the BioResource, but important to put this in context. So if it's a CF study, we would normally expect that this would also involve linkage to the CF registry, and that we would have some opportunity to oversee this. But they may get approached for non-CF studies. So for example, if they tick a box in terms of other comorbidities or polymorphisms. Now, most of those, if they happen, would be just data access requests or analysis samples that are already stored. It's much less common that patients actually get asked back for things, and the patients in the rare disease BioResource tend not to be used as controls anyway. The way they would be asked is the way they've identified they would want their want to be contacted. So it may just be a question of receiving an email and they don't have to do anything about it. And the number of asks they would get is capped at 4 per year. What we're not expecting is that CF patients will get deluged with a whole series of clinical trials that they will suddenly be asked to take part in. And the BioResource will be available to commercial companies. That's a strength. I don't think it's a weakness, but it's something that maybe patients need to be aware of as well. And we have an ambitious target. When we set out last year, we thought we would maybe get 6,000 patients over 3 years. The start has been slow for a variety of reasons, but we are very keen to push this and get as many patients in as possible, because this only works if we have large numbers of patients in the BioResource. The advantages, however, are considerable, and I'm about to run out of time, but I'm going to rattle through this quickly. We think this will establish a powerful resource for future research, and it's a quite unique offering. There's nothing like this in the EU. There's nothing quite like this really in the US, where we're recruiting across the whole nation. It's open to UK researchers, and for researchers in the room, I'm sure you can see there are many ways in which this could be helpful. It's supported by CTAP for setup, and it's a good opportunity to engage with patients when you're speaking to them about trials and research in general, and full credit to Exeter, who are the highest recruiting centre, and that's the team there. It's also supported by NIHR, so R&D departments do love this study, and they will be helping you set these up, and a very good opportunity for other researchers to get involved. So in Manchester, our PI is Cassie McNaughton, our very excellent specialist CF research nurse. It doesn't have to be one of the main clinicians. It can be another member of the team or a fellow. So additional resources, that's the BioResource website. That's the CF Trust BioResource with video. Me and Andrew's been interviewed by a patient who gives his own powerful story about how him and his brother had very different expressions of disease. Hannah Stark from BioResource is at the back of the room, I think, and is available later on. And there's more next week at the CTAP meeting, where Sophie and Hannah will also be talking. Thank you, and I'm sorry I've overrun. I think a minute ago.
It's okay. We knew it was happening. Thank you very much, Alex. That was terrific. So we've now got a presentation from Professor Alan Smith at the University of Nottingham. Unfortunately, he can't attend today, understandably, because he's in clinic, but he has pre-recorded his presentation, James Lind Alliance Priority Setting Partnership for CF Refreshed. So we'll roll the video now.
Hello, everyone. I'm sorry I can't be with you in person today, but it gives me great pleasure to talk about refreshing the James Lind Alliance Priority Setting Partnership top 10 questions for clinical research in CF. And I hope that this project will be just as refreshing as a cool pint in your local hostelry. Many of you will be aware of the top 10 questions for clinical research in CF, which we published through the James Lind Alliance and developed in 2018. I've shown them on this summary slide. I won't describe all of them, but the top 3, for example, are what are the effective ways of reducing treatment burden in CF? How can we best address gastrointestinal symptoms in people with CF? And what are the most effective treatment for infection with non-tuberculous mycobacteria? And the others I've listed for you there. This process was a collaboration between the patient and the clinical community, and it was supported by @QuestionCF, which was a bespoke Twitter feed. Just a few moments to explain the terminology. So the term James Lind refers to the James Lind Alliance, which is supported by the National Institute for Health and Care Research, or NIHR. James Lind was an 18th century ship surgeon, and he conducted the first clinical trial. And this was looking at treatments for scurvy, and he established that citrus fruit was an effective treatment for scurvy, and he's given his name to the James Lind Alliance. This brings together patients and clinicians across the spectrum of disease, all different disease areas, everything from mental health through to cardiovascular disease. And so far, there have been over 100 of these priority setting partnerships conducted. So you may remember how we went about our first James Lind Alliance. We did this through a series of online surveys. The first was to elicit questions, and it asked a very simple question of the respondent. What question about CF would you like to see answered by research? And we took free text responses. And we followed that with a prioritization survey where participants could prioritize their top 10 questions from a list of about 70. And we were absolutely delighted with the engagement that we had with these surveys. So we had over 500 individual responses to the first survey from 21 different countries, giving us over 1,000 different research questions. But perhaps what was most reassuring and pleasing was that half of these responses came from the patient community and half from the clinical community. So this really did feel like a partnership between patients and clinicians. And the whole process took us about a year last time. We started with setting up a steering group and finished with a final workshop. In between, there were 2 surveys, one which elicited questions on clinical research in CF, and the second one which asked the respondent to prioritize which were the most important questions. And we were funded for this work through the CF Trust, our own Nottingham Hospital's charity, and also the University of Nottingham. What was hugely important to the success of the first James Lind in CF was at QuestionCF, the Twitter account I mentioned earlier, which was set up by Zoe Elliott and drove engagement with our surveys and our workshop. One thing that was really critical to us as a group developing these top 10 questions was to understand whether existing research at the time that the James Lind Top 10 was published was reflective of the priorities of the patient and clinical community. So we did a piece of work where we looked at ongoing research at the time the James Lind Top 10 was published. This is research where the protocols were available in online registries such as clinicaltrials.gov and ISRCTN. And we found that only about a third of ongoing research matched one of the James Lind Top 10. Moreover, there were considerable differences in the number of research studies for different questions. So for example, the question about motivation to support adherence had about 25 ongoing research studies addressing that, whereas the question about treating non-tuberculous mycobacterial infection had only 2 ongoing studies. So not only were most ongoing research studies and trials not addressing questions which were a priority for patients and clinicians, but also those that did address those priorities, there was a disproportionate emphasis on some questions rather than others. We also did a piece of work where we wanted to find out a little more detail about the questions in the top 10, because some of these questions were a bit broad. So in a sense, we went back to the patient and clinical community and said, you told us this was important to you. Can you tell us a bit more about that? And we did that for the 4 questions that I've shown on this slide. What are the effective ways of simplifying treatment burden? How can we relieve gastrointestinal symptoms? The motivation and support for adherence question, and also the thorny question of can exercise replace chest physiotherapy? And we had some great engagement with these further surveys and some really interesting granular data from the patient and clinical community. I'd just like to highlight one quote that we had from a parent of a child with CF. And she said, there must be research on how to take things away. I hate it when new meds come out now because it means we just have to do more stuff. We need more things coupled together. We need treatments prioritized for us. And that really struck home. I hate it when new meds come out because it adds to the treatment burden. One of the key objectives for us in developing the James Lind Top 10 with the patient and clinical community was to bring new research funding into CF. So have we been successful in doing that? Well, following the publication of the James Lind Top 10, there were 2 commissioned calls through the NIHR. One was for the health technology assessment, and the second one was really innovative and hasn't been done before. It was a cross-program call. So it allowed applicants to apply for funding to HTA, efficacy and mechanisms evaluation, innovations for industry, program grants, and research for patient benefit. So a whole raft of research funding opportunities at different stages of the research pipeline, but all highlighting the James Lind Alliance Top 10 for CF. And one good example of a piece of funded research was the CF STORM trial, $1.5 million, led by Gwyneth Davies and Kevin Southern, and looking at that really important question of, can we reduce the treatment burden? So these were participants who were starting Kaftrio. Could they stop some of their existing inhaled medication? And this study has already recruited over 100 patients towards their study target. I'd also like to highlight some work we're doing. This is the gut imaging for function and transit in CF, or GIFT-CF series of studies. And so far, this has been funded to over $1 million through a consortium of funding, including the CF Foundation in the US, the CF Trust, and Vertex Pharmaceuticals. And then overall, the UK CF Trust has provided 10 million of research funding to support James Lind priority questions, and they've leveraged another 13 million to address these questions from other funders. So I would argue, yes, this has been a success, and yes, it has brought in new funding to CF and focused that funding on questions which are a priority for clinicians and patients. Well, this begs the question of why refresh the JLA Priority Setting Partnership Top 10? Well, 5 years have gone by pretty much since the top 10 questions were agreed. Some of the top 10 questions, as we've seen, are now being answered by large clinical studies. Many, but not all people with CF are benefiting from CFTR modulator treatment, and that may have changed their priorities. And furthermore, we've adapted to the pandemic. We're doing research in different ways, and also that might change what are the priority questions for patients and clinicians. So what's the plan? Well, we started at the end of last year. We hope to get this done in a year. We've already had our first survey. That was open from mid-January to mid-February. And in that, we asked respondents to choose 3 of the original 20 James Lind priorities. So we extended it, not just the top 10, but the top 20. And then we asked them to suggest 2 new questions. We've had fantastic response to this first survey. Over 1,300 people have responded from 25 different countries, suggesting almost 1,000 new research questions. Interestingly, around 3/4 are from the lay community this time versus about 1/4 from the professional community. And perhaps not surprising that people with CF and parents of children with CF were the top responders. Around 2/3 are on modulators, mostly for 1 to 2 years. So these are mostly people on Kaftrio. The average age of the respondent or the patient on whose behalf a parent was responding was 27 years. And around 2/3 of people responding were female. This shows the breakdown of the professional responses. So kudos to the dietitians and physiotherapists who really kicked ass here. I've shown the figures for the respiratory pediatricians, physicians, and so on. And we had responses from around the world. Just under half were from the UK. About 40% were from Europe. We were delighted with that response because first time around, 5 years ago, we had about a 10% response from Europe. So we're really pleased with the engagement we've had this time. Also, we've had good engagement with North America, Australasia, and other parts of the world. So we do think that our final refreshed top 10 will be representative and generalizable. For the second survey, we're going to ask respondents to choose from a list that will be compiled from the 15 of the top 20 last time, which were highest on the respondents list, and the 35 new questions. By the end of the summer, we hope to have a new top 10 list. We're going to share that widely. and then we will work with funders to bring further funding to CF research to address priorities which are important for people with CF and for clinicians. I'd like to thank everyone who's contributed to the James Lind Alliance Priority Refresh for CF. In particular, our management group shown here, one or two were camera shy, but most people are shown in this slide. I'd like to thank also our funders, the CF Trust and Research England. We've had terrific support from the James Lind Alliance for this project through the James Lind Lab. And this is the first time that a refresh of a top 10 priority setting partnership has ever been undertaken. So it's been very exciting to work in this experimental space that is supported by the James Lind Alliance. And of course, I need to thank Zoe Elliott and everyone who's been involved in setting up the @QuestionCF Twitter account and webpage, which has really driven engagement with the project. And I'd like to thank all of you for listening.
Well, huge thanks to Alan for pre-recording that. I'd like to introduce our next speaker, Professor Andreas Floto from the University of Cambridge, and his presentation, UK CF Innovation Hub. Over to you.
Thanks very much. Hi, everyone. Thank you to the organisers for the opportunity to chat. Okay, so I'm going to tell you in the next few minutes something about the CF Innovation Hub in Cambridge, which is, as Lucy mentioned, a strategic partnership between the University of Cambridge, providing £5 million to research and a further £2.5 million in research infrastructure, with the CF Trust that have already delivered £3.5 million of additional funding for Cambridge-based research. So this is above and beyond the research budgets and that it had before, and hoping to generate another £1.5 million commitment for UK-wide research. So really the thinking behind the Innovation Hub was what would be the outstanding big problems even with highly effective modulator therapy in CF. And really we kind of grouped these together to around the theme of precision medicine for CF, but specifically efforts on drug discovery, new antibiotics, new anti-inflammatories, regenerative therapy, that is to say cell-based therapy to rescue and repair damaged lungs and provide better models in vitro for drug testing, and smart technology, some of which some of you will have heard me chat about in other fora. But in addition to that, there was support for national networks to uplift and support the research infrastructure in the UK for researchers, and that included training, reagents resource, sharing, technology transfer, and the UK CF Biobank that Alex spoke to you about, and particularly our support for people with CF wanting to join the CF Biobank out of England, so Scotland, Northern Ireland, and Wales, that, as Alex said, aren't covered by the award from the NIHR. Okay, so we are in Cambridge. The Innovation Hub is a distributed network across Cambridge, and we have a physical centre now in the new Heart-Lung Research Institute. So this is a bird's eye view of the biomedical campus. Some of you will know, but this is the new Papworth Hospital here, and this is the new Heart-Lung Research Institute here. But we have labs distributed across Cambridge. And so as an idea of kind of bringing together this designated physical space, as well as to supporting the clinical research that we're doing at Papworth Hospital. So this is what the new Heart-Lung Institute looks like. It's just opened. We are. It was only 3 weeks behind schedule despite COVID, so they've done pretty well. And basically brings together kind of wet lab people doing experiments in test tubes and stuff with data scientists in order to kind of find innovative solutions to big problems. And we have a designated space here in green, which is the CF Research Centre. We're very excited about that. And so breaking down the aims of the Innovation Hub, it's really antibiotic discovery is our main thing. We do a bit of anti-inflammatory stuff, but it's antibiotic discovery. And I'll tell you a little bit more about that in some detail. Regenerative medicine led by Ludovic with help from Pietro and Emma, and the smart technology led by Hela van der Schaar and John Wynn. And again, I'm not going to talk about those two aspects today. Instead, I want to get kind of into the weeds of the antibiotic discovery, aware that there are potential organizations looking to co-fund this work. Me and Paddy are doing outbreak or breakout session, not outbreak. It could be an outbreak. We've had too much of that. So breakout session number B online, where we're going to have a fireside chat about all things about antibiotic discovery. And so for those of you online that want to tune in, we're going to have a kind of an introduction to the problem. The next few slides are really aimed at potential funders, and I'm just looking at Katie from LifeArc in the audience for no particular reason. Okay, fine. So our approach really is based on this concept that you can simplify a lot of the chemical diversity of drugs, which limits conventional high-throughput screening by breaking these drugs into Lego bits. So these are components that make up more complicated drugs. And if you do that, you can basically get away with screening less than 1,000 of these Lego blocks for low affinity binding to drug targets. And then by iteratively looking at what the structure looks like, changing the compound, growing it, or merging these fragments, you can then rapidly develop drug-like compounds. And this approach, which is called fragment-based drug discovery, was pioneered in Cambridge by Tom Blundell and Chris Abel, who sadly died a couple of years ago. But they were both heavily involved in the CF antibiotic discovery effort, and continue to, and in the case of Tom, continue to be so. But this approach has led to a number of drugs already approved by FDA and EMA for therapies, including for cancer. So it works very well. It's transformed drug discovery. And I just want to show you the kind of things that we can do. Is basically taking a structure, which you can derive either from cryo-EM or from X-ray crystallography. You can identify pockets of what are called unhappy water, that is to say hydrophobic pockets where water has to go because otherwise there'd be a vacuum, but doesn't really like it. And these end up being quite good ways of predicting where the initial fragments will bind and allow us to screen which targets are good and which targets are bad. And in fact, we can then map and grow the targets, the drugs, from one hotspot and grow that into another hotspot to generate these drug-like molecules. Okay, and this kind of works, all right? And we've showed this in a publication a couple of years ago where we did an initial screen, identified 2 fragments binding to this abscessus target. We were able to merge them to this compound, which showed efficacy against the bugs, not only abscessus, but as it happens, TB and leprosy. So we're kind of really excited by that. I guess we've progressed that target, which happened to be TRMD, with other targets, and now we have molecules that are at the kind of level of activity that we can stick into mice as the kind of ultimate preclinical go-no-go decision point, and then hopefully partner with pharmaceutical companies to take that to clinic. So we're really excited by that. But if I'm being honest with you, the vast majority of these campaigns end in failure. And they end in failure because of this problem. So here, this is the original fragment, and we can go through a series of chemical elaborations where you can monitor the affinity of the compound against the target reduces down to nanomolar levels. That's brilliant. But if you look at the activity against the bug, it's completely unchanged. And so it kind of tells us that there is a big problem that we haven't solved yet. And that problem is understanding permeability. And so you can imagine how some drugs can get in, other drugs can't get in. Some drugs get in, go out, and other drugs go in and get metabolized. And actually, we have no idea what this looks like, what the rules are for chemical permeation and retention. And that means that we are stuck with the problem of developing great drugs against targets in vitro, but they don't work against the bugs in real life. Okay, so what we've done is we've experimentally determined the chemical permeability properties of 10,000 or so compounds using a multiplexed mass spec method. And that allows us to map out chemical space to see what kind of properties drugs that get in have. And then we can train machine learning predictors to guide the chemist to say, right, well, we want a triangle, but then we've got to have a circle then, and then a hexagon, a pentagon after that. And so this kind of potentially allows us to get rid of that barrier and start generating antibiotics almost at will, which is very exciting. This is a kind of a cartoon, but this is what chemical space looks like. So you can imagine these hotspots of certain types of chemicals corresponding to methods of getting in to the bacteria. And again, we can explore that genetically from the bacterial side. Okay, so those that are interested, this is a mass spec method where effectively we can multiplex up to 38 compounds per well and then scale that up into 96 wells. That allows us to screen thousands of compounds a week and allows us to either use this method to ensure permeability all the way through the chemical elaboration process. We can develop predictors to allow us to screen in silico using computers and then chuck out all the ones that aren't going to get in, and also understand some of the basic biology of permeation. This is just showing you, these are green bugs before our lysis step, and this is the green bugs after our lysis step. All the green has gone out, and the PI, the red, propydium iodine, is allowed in. So this shows you the effectiveness with which we can permeabilize the bugs. And in fact, you can look then at the binding to the background and to the envelope of compounds, and also the amount that gets in. These are drugs that are in use clinically, bedaquiline and ciprofloxacin, but we can monitor the amount that gets in and also the metabolites of these drugs over time, which is kind of really exciting. It allows us to then compare one drug with another one and interactions between one drug and another drug. And obviously we can do that for compounds that we don't know anything about from our screen. And in this one, you can see this compound gets in great, but within 24 hours is chucked out with this development of this metabolite. And also, because we can inhibit efflux pump activity, we can then show what would be the amount that we got in if the efflux pumps weren't working. And so this is kind of really exciting at the moment. We're just ready to go to explore some really interesting biology. In the last few minutes, I just want to rush through some other approaches that we're using in order to get to effective antibiotics quickly. One other way of doing this is to what we call joint scaffold target discovery. So we take a diversity-oriented synthesis library. This is a chemical library where all of the compounds are radically different from each other, and then identify that. So we screen phenotypically and then identify the targets either using an arrayed CRISPR library or cross-linking using photochemistry or looking at escape mutants, or this whole cell thermal shift assay allows us to detect not only the target that we think is being involved, in this case this one, but across the whole proteome of the bacteria. The other thing we can do is use forward genetics using CRISPR technology in a pooled way to identify all the essential genes, in this case in abscesses, and also their individual vulnerability. Using CRISPR-I, we can tunably knock down the targets of these specific essential genes, and then do more computational biology to work out which binding pockets are resistant to mutation. That's to say, which binding pockets are likely to resist being mutated out. We've used quite a lot of advanced reverse genetics, which we've published recently last year in Science and Nature Microbiology, to identify genes controlling virulence. This is the network in abscesses, and also some very exciting systems level genomics using machine learning to encode genomes as protein graphs. And then finally, we think we're in a position now to apply machine learning to generate in silico screening platforms where we take billions of compounds. We can then screen them against the structures in the computer. We can then filter out the ones that don't bind using our permeation model, and end up solving this problem of which drugs can bind and get into the bug all on the computer. We're not there yet, I have to say, but this just shows you where we've got to at the moment. So this was screening 10 billion synthesizable compounds. we were able to screen and then dial up you can buy the compounds that are hits and then solve the crystal structure. And you can see the original crystal structure here in white compared to the prediction in blue, and the same here in white is the actual one, and the prediction in yellow. So this is kind of unprecedented accuracy of in silico screening. And now we're hopeful that we can combine that with the ability to filter for permeation. Oops. Very penultimately then, we are very excited by this nanodrop technology. This is water in oil drops, and that allows you to take chemical libraries or stapled peptides, merge them with bacteria, incubate them to see which ones prevent growth, sort them, and then split either for DNA sequencing in terms of encoded libraries or mass spec to identify the compounds that are working. So we're very excited and working with some folks in chemistry in Cambridge. And then finally, opportunities to use nanobiotics, which we did a few years ago. So these are taking compounds and making them into polymers to then generate nanoparticles, and then using those to treat mycobacteria. This is fish, and these nanoparticles get taken up by macrophages, go to the site of infection, and kill the mycobacteria. And so now we're in the position where we can do the same thing for lots of toxic drugs, but which are still very effective for mycobacteria, stick them to antibodies that detect the bacteria, and generate new antibiotics that are potentially rapidly introducible into clinical practice. So I'm going to stop there. Thank you all for your attention. To plug the breakout session B after lunch, and then just to thank the CF Trust for their support, as well as the University of Cambridge. Thank you very much.
Thank you very much, Andres. So we've got our final presentation now, and then we'll move into the Q&A. So I'd like to introduce Dr. Paula Sommer from the Cystic Fibrosis Trust, and Dr. Bev Isherwood from the Medicines Discovery Catapult, and their presentation, Partnering for Impact, the CF AMR Syndicate. Thank you.
Thanks, Catherine. So I don't have to tell this audience that antimicrobial resistance really is a problem for all of us. In January this year, The Lancet published a report that estimated that AMR killed more people than HIV, AIDS, or malaria in 2019. And the report also showed that Pseudomonas aeruginosa, already a World Health Organization critical pathogen, was one of the top 6 pathogens associated with these deaths. So we really come to this point because of a significant lack of investment in the development of new antimicrobials. And there are lots of reasons for this, but primarily is because of the inability of industry to recoup their investment costs. So the world's starting to recognize this, and there are numerous incentives to preserve our existing antibiotics, and also various push and pull incentives to promote the development of new antimicrobials. So what does this mean for CF? So we know people with CF are susceptible to lung infections. You've all seen the snapshot from the UK CF registry reports that show that the lungs of people with CF are colonized by a variety of different microorganisms. Almost 40% of adults are chronically infected with Pseudomonas. So we're really in the position where we're in a very fortunate position that there are CFTR modulator drugs. But while we know the short-term clinical effects of these drugs, the longer-term effects, particularly on infection, are not that well understood, and there's some conflicting reports in the literature. But what we do know, so far anyway, is that infections are not eradicated. And of course, we have a population of people with CF who are unable to benefit from these modulator drugs. And those already with significant established lung disease. And so I think we can all agree that antibiotics remain a key and necessary therapeutic for CF. And so there's really an urgent and unmet need for the development of new antimicrobials and improved diagnostics for treating infections in CF. So the Trust recognized this and seized the opportunity to partner with Medicines Discovery Catapult to form the CF Antimicrobial Resistance Syndicate. Now, the mission of the syndicate is to accelerate the translation and adoption of new CF antimicrobials and diagnostics to the clinic, and hopefully through collaboration and doing this so faster. So the unique value of the syndicate really lies in a deep understanding of the needs and priorities of people with CF, enabling the development of guidance, of support, and of access to funding. So the syndicate members are drawn from the CF and wider community to provide this deep understanding.
Yeah, so the approach we've taken, as Paula says, is to work with everybody in the research community and beyond that. So the first thing we did when we formed in 2019 was to pull together a cross-sector group. So researchers from industry, clinicians, academic researchers, people with CF and their carers. And we came together really to understand and identify areas of patient need, priorities, then leverage that cross-sector expertise that we all collectively brought together to initiate and catalyse new drug discovery efforts, R&D efforts, keeping the patient needs and priorities at the centre of that. And so what we did first as a group was to come together, really look at the current state of the pipeline of therapeutics in development, antimicrobials in development for CF, and also engage with lots of people involved in the field to understand the key challenges that are being faced in progressing those from start to the clinic. And we focused on two challenges that emerged from those workshops and discussions and data analysis. The first is that there isn't actually a well-established and agreed preclinical roadmap to take your compounds right through from the start into the clinic. And in some cases, actually, there aren't really good preclinical models to help select the best compounds. And the second challenge that was identified through this exercise was the difficulty researchers face in accessing clinical isolates, pathogen bug samples that are relevant for CF. And so what we then set about doing with the community was to put in place a whole suite of R&D, collaborative R&D projects to address these challenges. And on the right of the slide here, you can see the list of projects that we have. And Paula will take you through a couple of those in a bit more detail now.
Yeah, so as Bev says, one of the enabling resources in the toolkit was the development, or is the development, of the UK CF infection biorepository. So the biorepository was really set up to ease access for both academics and industry to CF relevant strains and isolates sputum samples. The biorepository virtually links up some these already existing collections of highly well annotated and high quality CF clinical strains and isolates and sputum samples, along with the extensive expertise of those centres. So the biorepository is virtual. There's a single access point which allows for easier advertising but also for easier access. So then also to address one of the major barriers to drug developers in this space, that is of the lack of a well-defined preclinical pathway, the CF Trust and Cystic Fibrosis Foundation co-funded an SRC led by Joe Fothergill in Liverpool. And this is around developing an evidence-based preclinical framework. So Dan O'Neill is one of the PIs on this SRC, and he'll talk about this a little bit later. And then the syndicate also produced guidance for drug developers in the form of target product profiles for CF-related infections. So these TPPs are really guidance documents that highlight the key characteristics and requirements that CF antimicrobials should meet in order to address the needs and priorities of people with cystic fibrosis. So we produced these TPPs in an iterative process, but really turned it all on its head and started off with focus groups of people with CF. And from these focus groups, we developed a patient-focused generic TPP that really sets out the beginning of the document and feeds into the specific TPPs around the development of antimicrobials for Pseudomonas and Mycobacterium abscessus infections.
So what's next? We've established ourselves, we've built and continue to build a suite of tools for the community to access and continue to develop. So where are we headed next? And it's really in three main areas. So we do want to continue developing the tools we've described, but also to start to build additional areas of need that are identified by yourselves and others. to help overcome challenges in antimicrobial development, but also diagnostic development as well. And so this specifically is a new project we're just starting now in partnership with LifeArc and also the NIHR up in Newcastle, the In Vitro Diagnostics Collaborative. This project will develop a target product profile around the needs of diagnostics to enable us to better manage lung infections in CF. And we're looking to build over time more and more different enabling tools as are needed. The second thing we want to continue to focus on is over the past 2 years of operating the syndicate, we have built a really great cross-sector network of people who have really helped guide us and support us in our efforts here. We would like to formalise that network and create a unique community of what we're going to call the CF AMR network, open to everyone. And really what we want to use this network for is to help people to facilitate knowledge exchange and also spark new collaborative opportunities. And Paula will say a bit more about how to get involved in that later. And then on the right here, we want to start looking more proactively at how we support that emerging pipeline and really accelerate the existing pipeline and add more antimicrobials to it, new diagnostic approaches to it. And so in order to do that, what we really are keen to do is to help support researchers, innovators operating at all different phases of the drug discovery pipeline or the diagnostic development pipeline. So right early on in academia, but also new startup companies entering the CF space, established companies, large pharmaceutical companies. And to do this, it's really great that we're continuing to evolve the syndicate, and we're joined in a partnership alongside MDC and CF Trust with LifeArc. And together, what we want to do is to put in place some specific initiatives, and I just flag a few here to help address the specific needs of these innovators along this drug discovery pipeline. So at the left-hand side, what we're really keen to work on this year, especially playing to the strengths of LifeArc in helping to support translational efforts from academia, is looking for collaborative discovery programs with academic labs that have got the latest new targets, new approaches, and align those with our target product profiles that we know will meet the needs of people with CF. A bit later on down the pipeline, we're really keen to work with new startups, emerging SMEs to incubate and accelerate their ideas and thoughts, put them in contact with the right expertise, funding, specialization. And then down towards the right is really making sure that people have got that line of sight of those new innovation opportunities coming through to facilitate partnering. So just to show you one live example of one of these initiatives at the moment, this is a program that we have running to help incubate new startup companies that are entering the CF antimicrobial space. We're benefiting from a program that's run in partnership between MDC and an organization called WeShare, and they have an impact-driven revolving funding model which they haven't previously applied to early stage medicines discovery. This is a new pilot for them and everybody. And what we hope to do here is to identify promising startups, actively then raise philanthropic funding for those projects. WeShare would provide that to the startups as a grant to accelerate their work, and then ultimately recycle that funding back into the next generation of molecules being developed. So the stage we're at now is we have 3 promising CF AMR startups, really exciting early stage therapeutics that have been selected through this program. They're going to receive some bridge funding, and what the syndicate is doing is to really incubate those ideas and connect them with the right tools, expertise, small amount of bridge funding they need to get them to the next stage. So that's just an example of where we're working in partnership with other organisations to bring that whole package together for people. And then I will hand to Paula to talk a little bit more about the CF AMR network and how you could get involved. Yeah.
So we really built the syndicate for the benefit of all the stakeholders in CF antimicrobial drug discovery and diagnostic development, right from people with CF right to pharma. And to make sure that we really address the needs and priorities of all of these people, we decided to formalize and form a CF AMR network. So through the network, we aim to share all of the resources that we have developed and are developing. We hope to promote collaboration and then hold various events, workshops, and focus groups that are really defined by the community. It's open to everybody who wants to join, and it's free to join, so please do so if you want. So we've given you a really whistle-stop tour of the syndicate and our activities, and it just remains for me to thank the members of the Syndicate Steering Committee, some of whom are in this audience, for giving so freely and generously of their time and expertise, and of course also the wider CF community who have helped us develop all of these different resources. Thanks.
Thank you very much, Paula and Bev. So we've had a fantastic series of presentations. Thanks to all of our speakers. If Andres and Alex, do you want to pop up back on the stage? We've got time now for some Q&A. So we're going to take the lectern away. And let me just explain how it's all going to work. If you have a question for our speakers, whether you're online or in the room, if you can say who your question is for, that will be really helpful. We've got a roving mic in the room that Jacqueline is going to be commandeering. And Belinda has an iPad, so she's the in-room spokesperson for the online delegates. And what I'd like to do is to alternate between a question online, a question in the room, just to keep it fair. And I think for this session, I'm going to start with the questions from our online delegates. We'll get one of those, and then we'll come to the room. I'll let you know when to put your hand up. So I hope that all sounds okay. Belinda, any questions? We need the mic down here, Jacqueline. I know no one's looking forward to having their questions answered, but hopefully that has now changed. So, Belinda, any questions from our online audience?
Yes, we do.
Great.
We have a question. Is this microphone on?
Yeah.
Sorry. We have a question about the bioresource. Somebody's asking, can they take part in the bioresource if they're taking part in a clinical trial?
Absolutely. This isn't on. Absolutely they can. It's observational study. It's just a sample of blood taken at one time point in the first instance. And that's all that's required. They can take part in any other number of studies as well.
Great, thank you. So let's go to the room next. If you've got a question for one of our speakers, if you can put your hand up and Jacqueline will come over. Okay, I'm going to give you a bit of time to... Oh, thank you, sir.
I'm not sure...
It's on.
Oh, it's on, yeah. Not sure Sir's appropriate. It's Paddy from Cook. I've got questions for everyone, but I'll just ask this one to Alex. Just with the potential modifier genes, you've explained Gary's work really nicely there, that study. But are there any of those genes where you could potentially activate one of them or inhibit one of them and really bring about a clinical benefit for a person with CF?
So that's a million-dollar question, isn't it? And there's certainly theoretically, there are other ion channels that could be activated or inhibited that may impact upon the final expression of CF. There's other pathways that could also be that we think already are overexpressed or underexpressed in CF. And if a strong signal is obtained from work like this, then that would drive innovation and drive people to look at molecules that are out there or develop molecules for those pathways. So it's entirely complementary to the kind of work that Andreas is doing.
Okay, so we'll go back to online for a question. So we'll go to Belinda and then we'll come to you next.
Hi, this is a question for Paula and Bev. Do you know, are there any events that are happening with the CF Syndicate in the near future?
Yeah, we are planning a series of events for the year. We're very much going to be led by what the community would like. So if there are workshops and things you feel will be really beneficial, do let us know. The one that we are planning will be for October time, and we're planning a joint event between the UK CF Infection Biorepository and Pipe CF, and that will be a very interactive workshop focused on some of those key challenges around the preclinical pathway and accessing clinical strains and isolates.
Okay, thank you. We've got a question here in the room.
Hi, Margarida Amaral from Portugal. I'm catching up on Patrick's thoughts about modifier genes, which you are sort of discovering with this bioresource. As a follow-up to that, I think it would be really interesting to actually do some sort of screening regarding the role of those genes, you know, regarding CFTR or any other parameter that you'd like to assess in respect to CF. So as to maybe some of them are potential drug targets. So I think this would be very interesting.
Absolutely, and the BioResource, all the BioResource does is collect that information. It's up to the research community then to come up with those studies and, if necessary, the funding to analyse that. But the first thing we need to do is get as many patients as possible into it.
Great, thank you. So we'll go to Belinda next, and then I can see there's a question at the back. So Belinda, any online questions?
There's just a follow-up question about the syndicate. Paula, you mentioned about people being interested in joining the network. Can you say a bit more about how they join?
Yeah, sure. There's there's an email sign up to it, right? Yeah, so it's on our website, and our website is almost ready to go. So it's not quite ready to go, but when it is, obviously we'll advertise it and promote it.
Great, thanks. We've got a question at the back of the room there.
Yeah, hi, it's a question for Alex again. Alex, I'm quite embarrassed.
I'm getting hammered by this audience.
We're one of the sites that's been very slow to get started, not because of lack of enthusiasm, but just because of logistics. And we're doing another study at the moment that sounds like nothing. It's just a blood sample, but it takes a clinical fellow all day to get like 4 patients in and data, etc. Is there anything within the ethics that would allow us to just mail shot the patients, to send them all the leaflets and all the information sheets? Because I think a lot of people would want to do it. Mm-hmm. It's just that if they could bring that form back in with them, sign it, we could have a sense check conversation. We might be able to get much more people much more easily for the cost of a stamp.
That's a very good question, Jane, and I don't know the exact answer to that, but Hannah, and you better run now, Jacqueline, all the way to the other side of the room. Hannah in the far corner over there might know the answer to that. Hannah, is it okay if I send that your way? Thank you.
No.
Jane, you better not have another question again after this.
Hi, yeah, the short answer is yes, we can do that. If you want to find me, I'll explain how, is the very quick answer. But yes, we can do that with a little bit of discussion on the initial letter, but it's entirely possible and within the ethics.
And the other important thing is CTAP. At the CTAP meeting next week, we're going to go through some of the practicalities in a bit more detail. So that's something we can pick up then. Thank you.
Okay, thank you. Belinda, I'm so sorry, Jacqueline. Is this a full-on workout? No, okay, no questions online. So hand just here, and then we'll come over to you next.
Hi, I've got a question for Andreas.
Oh, thank goodness.
I enjoyed your talk. There was quite a bit in there. The antibody-coupled drugs. Could you tell us a little bit more about how that would work, and then, do you have any of those that are already functioning?
Yeah, so that's a very good question. So I guess antibody conjugates to antibiotics have been shown in proof of concept studies. There was a very famous study with Staph aureus a few years ago in Nature showing improved activity, and the expectation is in that sense, in that example was that the antibiotic was cleaved in an acidic environment, so it would bind to the bug, it would be internalized, and then it would be acid cleaved. What we've been developing are methods to link the antibiotic or the nanoparticle to the antibody with a linker that's only cleaved by the mycobacteria, so using a beta-lactamase cleavable linker, which is very exciting because in principle at least, we should really minimize systemic exposure and only activate the drug when it's by the bug. Do we have any evidence that it works in vivo yet? No, we don't, but that's where we're going. So.
Cool, thank you very much.
Okay.
Great, thank you. So let's go to another question in the room, because I saw a hand up just over on the left, or my left, your right, and then I'll come back to Belinda for the online questions.
Thank you. Thanks for very fascinating talks, particularly development of antibiotics.
Do you want to lift the mic up a little bit?
Sorry?
If you can hold the mic a bit closer.
Oh, sorry. Is it too loud?
That's better. Go for it.
Thanks for some very interesting talks, particularly on antibiotic development. But really, there was a complete showstopper at the beginning when, and it's a question for Paula Stomer, that really drug development isn't taken up by companies because there just isn't the cost benefit. It's difficult for smaller companies. Regulatory bodies ask for huge studies, and as soon as you've got a major new class of antibiotics, they say, can this work in the community and so on, and then you get it registered, and they say you can't use it in the community, it has to be reserved for just a few patients. It's a ludicrous situation, which is been there for a number of years. COVID has one advantage that people have re-looked at regulation, conditional regulation. Rolling assessments by the MHRA, Kate Bingham has highlighted as one of the key things that help this country. Can we now move on, and is there now pressure to look at the registration of these drugs in a fairly radically different way, not irresponsibly, but looking at conditional release, carefully monitoring these patients who are critically ill? Is there any pressure from the various groups involved today, but particularly Cystic Fibrosis Society? Is there any lobbying going on through government, through the MHRA, to achieve this end? It was perhaps Paula Stoner might answer first, I don't know.
I can
Yeah.
take a start and then
Yeah.
you guys can chip in. So yeah, you're absolutely right around the challenges around the regulations and approvals of antibiotics. I often hear antibiotics talked about as a fire extinguisher. Actually, you want to pay for them, but you don't really want to have to use them until you absolutely have to. And so the economics of that is really difficult. But in actual fact, the UK is leading the way on pricing reform for antibiotics and has just completed a pilot with both compounds from Shionogi and Pfizer, which lead to value-based pricing of drugs rather than how much you're going to use. And so I think our hope across, and this could be followed by the Pasteur Act over in the US and also European equivalents, that if that continues to be adopted, it could start to change the economic value of investment that comes into antibiotics. So that, I guess, addresses a little bit to your point, but you're right. There are bigger questions around the stewardship as well and the best way to approach that.
No, I think Bev's addressed it quite well.
Maybe I can add something then. So I think there on the lobbying side of things, there certainly has been quite a lot of activity from the CF Foundation in the US to the FDA to look at this temporary or qualified licensing of drugs, particularly for diseases or infections where there's no other options. And I completely agree with what Bev's saying. So the ceftaz-avibactam is potentially a game changer for the UK, and I think is kind of the first, I would suggest, that the first evidence that pressure from a whole host of individuals, but particularly Sally Davis, on payment for registration rather than on a per use basis is actually gathering traction. So I think it's very exciting. So.
Great, thank you. Let's go to questions from our online delegates. Belinda, what have you got?
Hi, we've got a few. The first one I'm going to ask is to Andres. There's a question, I guess is inevitable almost, is what is the timescale for these antibiotics that you're developing at the moment before they can actually reach the clinic?
So of the two things that we're doing, the first is developing antibiotics ourselves. Unfortunately, that is for those lead targets, that is a long process, right? So we're at the point at which we're doing preclinical animal studies, which would be the kind of entry point into the fairly long period of evaluation for human use. So that is quite a long time traditionally, and I don't think there's any shortcuts to that. I think in terms of impact, though, probably the major impact is our ability to overcome the hurdles of antibiotic discovery in the ways that I described in my presentation, but particularly around permeation, should almost trivialize the development of antibiotics in a structure-guided approach. And if that is the case, if we can demonstrate that, then I think that's going to have widespread impacts for industry and academic groups.
Thank you. Let's take another question from online.
So this is a question about the James Lind priority refresh. I know there are colleagues in the room who might be able to take this question, so perhaps Catherine, you can make eye contact with those people. So the question is, how will research address these questions? How will research to address these questions be coordinated globally and ensure all areas are investigated?
So Nikki, hello. Do you want to talk to that one?
Hi, I'm Nikki. Oh, sorry, very loud. So last time we worked really hard to make sure we shared the results of the priority setting partnership as soon as they were available online on Twitter. We obviously published them through traditional methods in papers and things like that, but we also just basically told everyone we knew everywhere about them. We worked hard, Alan and I had meetings with the NIHR to highlight what the priorities were and to really push forward them as an important area for future research, and we will do exactly the same again. We're working very closely with Lucy here, sorry, and I'm sure you'll say the same, that you'll work hard, and we've got international collaborations on the steering committee and close connections, and we'll make sure that we just really work hard to make sure everyone in the world knows about them, and everyone will probably get very bored of us talking about them.
Thanks, Nikki. And Nikki and Sherry are doing a breakout on this exact topic during session 3. And just to say, because you saw my picture up there, I'm involved with the James Lind Alliance as a facilitator, and the setting of the priorities is the end of the beginning. The translating of those priorities into tractable research questions and engaging funders and researchers is the next stage. And the team and I have been incredibly impressed by the determination and commitment of the CF community, and I think we all feel very optimistic about the next steps of that project. Okay, so we are due to wrap up in about 4 minutes. Although I have just been red carded, but I think we've got time for a couple more questions. So we've got a question here in the room. You can pass the mic along.
Hello. My question is for Andreas. Really fascinating talk. So many amazing new techniques for discovering antibiotics. Are there any, in your opinion, that you think seem like they'll be more feasible in producing new antibiotics, or any that are much closer to actually discovering a new antibiotic?
Okay, so great question. So I guess the efficiency of fragment-based drug discovery is amazing. So you can go from purified protein, crystal structure, and then an initial fragment hit to a compound with some nanomolar affinity in weeks. I mean, that is incredible. And as I said, I think in the talk, its applications to cancer have been incredible. I think there are now maybe half a dozen FDA-approved cancer drugs which have just been made really quickly and effectively with this. So I think if we're able to solve this problem of permeation, then I think we'll be sorted, and that will allow us to almost at will take a target and then make something for it, and then that is really exciting, and I think that's likely to work. There's always challenges in identifying not only which targets are essential, but how much of them do you have to inhibit to kill the bug. This idea of vulnerability is actually very poorly defined. So often, we just need to try and sort that bit out as well. So, yeah.
Great, thank you. Belinda, were there any more questions from our online delegates? One more, let's make it a short one, and then we'll wrap up and we'll have a break.
So this is a follow-up to the question about clinical trials for participants in the BioResource. They were asking whether the 4 studies that you talked about, so each patient would be, or each participant would be eligible to take part in 4 studies afterwards, would they be observational studies?
So at the moment, that's all that would be likely to come out of that. It's not you'd be expected to do 4 studies. You would never be asked to do more than 4 studies. The reality is that the number of asked will probably be much lower than that, and it's going to be fairly infrequently that patients would have any further contact from the BioResource, but it will be capped at no more than 4 per year. But it's unlikely they would be, highly unlikely to be clinical trials. There's nothing that's sort of coming forward that's likely to occupy that space probably for many years, but observational studies or maybe additional samples or analysis of their samples.
Great, well thank you very much everyone. I'm going to draw session 1 to a close. So thank you once again to all of our speakers.